Välkomna till presentationen av Aroseniusfondens forskningsanslag för 2025. Jag heter Mikael Walter Litander och jag är ordförande i förbundet Blödarsjuka i Sverige och i Aroseniuskommittén som det utser mottagare av anslaget. Aroseniusfonden delar ut anslag till forskning som vi presenterar idag och även till resor, presentationer, deltagande i vetenskapliga konferenser. Det vi ska höra idag, det är nyheter om genterapi för hemofili A. Och det finns frågor och svar, Q&A, längst ner i bild. Om man vill ställa frågor, det går bra att ställa frågor på svenska och på engelska. Föredraget kommer att vara på engelska, men jag kan översätta frågor som kommer in på svenska. Welcome to Yamen Fu, postdoctoral researcher at Karolinska Institutet. Congratulations to getting the grant from the Arosenius Fund this year. And we are happy that you had time to present your coming research. We are eager to hear about it.
Thank you. Thank you so
Mm.
much for your introduction, Miguel.
Mm.
Good evening, everyone. I'm Yawen, and I'm a postdoc researcher in Karolinska. And first of all, I want to thank Arosenius Foundation, not only for the money, but also for the support and the honor. I really appreciate. And also want to thank our audience for your time. It's a great pleasure for me to have this chance to show my research. I'll talk about my research. So let's get started. So today I'm going to introduce my research about an easy one-step gene therapy for hopefully curing hemophilia A. So as we all know, like if an injury happens and causes bleeding in a healthy human body, and then there will be blood clotting as a wound to stop keeping bleeding. So actually the blood clotting is made or achieved by very complex coagulation cascade. As you can see, I show a figure here. So many different coagulation factors need to work together and maybe interactive together and then make this blood clotting. So why do we say this is a coagulation cascade reaction? It's like, for example, one coagulation factor need to be activated first, and then the activated coagulation factor can activate the next one, like downstream. So this reaction is like step by step. So that means like either of the member, like either of the coagulation factor has some problem or doesn't work, then it can cause bleeding disorders. So like the most two common types of bleeding disorders are hemophilia A and hemophilia B, which are caused by the lack of factor VIII or factor IX separately. And also like in our real life scenarios, there are always like more hemophilia A patients than the other hemophilia types. So hemophilia A affects more males than females because this is an X-linked recessive genetic disorders. And also people have found so many different types of mutation, like for the hemophilia A patient's genomes. So maybe we can try to understand and find out the like biology science background. Sorry, this one goes. Biology background or reasons for to explain why there is a higher incidence for hemophilia A. So the key point is factor VIII coagulation factor. So the factor VIII protein is very large and it has a very complex structure. So compare with other coagulation factors, factor VIII is like, you can tell from the protein size is way larger than the others. So that means the DNA sequence of the factor VIII is way longer than the other coagulation factors genes. And we name this gene is F8. So this is a gene DNA sequence of the factor VIII. So that maybe explain why so many different mutations for the hemophilia A patients, because this gene is complex and long, so it's easier to have some problems. And also because the factor VIII protein is very big, so it's easier to be recognized by the body's immune system. So for example, if some patients get the replacement treatment, it's like the injection of factor VIII product, and then like year by year, the patients might have the antibody of the factor VIII and then cause the treatment failure. So because the hemophilia is a genetic disorder, so it seems like gene therapy can be the only hope for curing hemophilia. And this one, this figure is like the history or development of hemophilia treatment. So at the beginning, people inject fresh plasma or some factor VIII product. It is very expensive, and also the patients need to get the injection every now and then. And then later, as we mentioned, there might be some antibody in the patient's body. So then the treatment is like cause a failure. And then later we also have some antibody treatment, which is also very expensive. And as the development of gene editing technology, so more researchers are focusing or give hope to the gene therapy, because gene therapy, we can probably like fix the gene mutation for the DNA, which is like can probably cure the problem from the root. Here is an example for hemophilia B gene editing or gene therapy by adeno-associated virus vectors. So the researchers, they put the factor IX gene sequence here, and then packed it into this virus vector. And then after delivery of the vector, and then the virus vector goes to the liver, and then the liver cells can produce the factor IX gene here. So hopefully the patients can have the factor IX protein in the blood. But as we mentioned before, factor IX is way smaller than factor VIII. So if we want to use the same strategy for hemophilia A to like fix the factor VIII, then it might be a problem, because the limitation of the AAV vector is very small. So the capacity just limited the package of factor VIII gene. So and another point is like this part of the DNA cannot be really integrated into or inserted into the cell's genome DNA. So like after a while, then maybe the AAV vector gonna be lost. So the coagulation factor that we delivered can be disappeared. So if we really wanna cure this problem, so we need to like insert our factor VIII gene into the genome. Recently we have not very recently, but now we have a very robust tool for gene editing, which is CRISPR-Cas9. Maybe you have already heard about. So this gene editing system simply work like this. To make this gene editing work, we need a Cas9 protein. So you can just understand this one as DNA molecular scissors. So this Cas9 protein can cut the double-stranded DNA, which can be our human genomic DNA. And then at the same time, we need a guide RNA. So this guide RNA can combine with this Cas9 protein and then direct this Cas9 protein to the place we really want to target. Because you cannot just cut a random place on your genome sequence. It might cause a problem. So with this simple system, we can direct the Cas9 to cut basically almost like wherever we want on the human genome. And also at the same time, if we give the cell a donor DNA, it's like a DNA sequence. So the red part, we can replace it to a gene of interest. So for our example, it can be factor VIII gene. So with this DNA template, and then after the cutting of the Cas9 and guide RNA, the cell can just repair its own DNA according to the template we gave to it. So after the editing, the new DNA gonna precisely insert the gene of interest, or for example, can precisely insert the factor VIII gene at the place we want. So hopefully the cell can permanently have this gene expression. So we know now for precisely insertion of factor VIII gene, for example, we need at least 3 components: the Cas9 protein to cut, and the guide RNA to direct the Cas9 protein how to cut, where to cut, and also a donor template to let the cell correct its DNA according to our design. So you cannot just inject the protein, DNA, RNA to mice or patients. It won't work. The blood or whatever body can clear this component. So we need to pack them into a nanoparticle, for example. You can just imagine we need to put some drugs into a capsule, and then the capsule can carry them to the place where we want to release them, then to deliver them where we want them to be. So during my postdoc research, we developed a very easy all-in-one delivery vehicles. This one is like virus-like particles, which can pack all the 3 necessary components we mentioned before. So we can pack Cas9 protein, guide RNA, and the donor DNA for example, for factor VIII in one single nanoparticles. But you don't need to be scared if I mention virus. You can just imagine if the virus can make us sick, that means it can infect human beings' cells, and then it's deliver the disease-causing things into the human cells. So we can do some bioengineer for them to remove the disease-causing part, and then we can replace them with, for example, therapeutic DNA, RNA, protein, or some drugs. Then we can use them to deliver the drug or whatever things we want good for the cells. So it's like the virus-like particle. And then I can easily just add this virus-like particle packed with all the necessary stuff for gene editing into the cells. For example, if I want to test in the lab, it can be a cell line or whatever. And then in the recipient cells, the cell's genome can be edited to the results I want or according to our design. Because I need to do a lot of tests in the lab, so I make an example here. I just knock in or insert a green protein to a gene locus. So only if the cell can repair the DNA and then insert the green protein to the place I want it to be, 100% correct, then it will turn the cell to green. And then in this way, I can determine the percentage of the green cells, and then I can know my gene editing efficiency. So we did a lot of optimization, and now we got very good gene insertion editing efficiencies in the cells. So the next step is to replace the green protein to something real useful, functional. So here will be our factor VIII. And like factor VIII in plasma, originally they are generated by liver and then can be secreted into bloodstreams. And there is another highly expressed protein in liver, which is albumin. So we can target albumin locus and then add the factor VIII after the albumin. Then hopefully the liver cells can produce albumin and factor VIII with the same DNA strand. Then hopefully the cells can like permanently produce factor VIII by themselves. This one shows like a workflow or my procedure or method to do in the lab. So first of all, I need to make the virus-like particles that packs all the necessary stuff we need for hemophilia A treatment. So it will be a Cas9 protein to target the genome, RNA, guide RNA, which target albumin to direct the protein to cut the albumin sequence, and also a DNA template donor with factor VIII DNA sequence to insert this part to the recipient cell's genome. So first of all, I can test in a cell line. I can just easily like add all the virus-like particle to a recipient cells or cell lines. And then after the cell culture, I can harvest the cells and do some, for example, flow cytometry measurement to see the gene expression, then I can know the percentage or like the efficiency of my gene editing system, and then maybe do some optimizations according to my results. And also I'm gonna harvest the cell's DNA, extract the cell's DNA, genome DNA, and then do some DNA sequencing, because really want to make sure the editing results are correct, and we don't want to introduce any more like extra mutations. So always need to be careful and double check. And then later we can do some animal experiments. So if we have hemophilia mice model, then maybe we can inject the mice with my virus-like particles, and then we can keep tracking the mice's, for example, the blood clotting function or other analysis. And if we have a wild type mice model, then maybe we can inject some virus-like particles and then sacrifice the mice and then get their tissue. So mainly gonna be liver, and similarly do the test for gene expression and also the DNA sequence to make sure the gene editing results. So our first aim is focused on the all-in-one delivery vehicle, the virus-like particles, because we always want to improve the efficiency as much as possible. So I did a lot of optimization for this part so far, but it's still going on because we always want something better. And I did a lot of optimization for the like capsid, the envelope protein, or the loading protein for Cas9, whatever else. And we have better and better gene editing efficiency now. So if we have a better editing efficiency, that means like in future, maybe we can use a lower dose for patients. So this is also like a safety concern. And also, of course, our final aim is to like achieve some gene therapy for hemophilia A. So we hopefully can insert the factor VIII gene in vivo, so the cells can have factor VIII continually. And these are some data from one of my PhD work, but this is made by some like hydrodynamic injection for naked DNA in mice. So there were a few mice like kept the expression of factor VIII gene for 1 to 2 years, which is basically the lifelong time for the mice already. And also like if we can recover the factor VIII or the factor VIII activity, for example, more than 1% or 5%, then hopefully the severe hemophilia A patients' life quality can be removed. So you don't need to make it to 100% activity. I think that's it. And at the end, I want to thank you, Aroseniusföreläsning again, and also thank you for our audience for your time. And I also want to thank my research group and our collaboration groups. It's very happy to work with my colleagues together in Karolinska, and they give me a lot of support. Thank you. I'm looking forward to hearing any questions from you.
Thank you, Yawen. I remind the audience that you can ask questions in the Q&A at the bottom of the screen in Zoom. You can ask them in Swedish or English. I have a fundamental question. So AIDV and AAV,
Mm.
are they both delivered with viruses? I don't quite follow the
Yeah,
difference.
I'm sorry for
Mm,
the short or abbreviation. The AIDV is actually a name we give to it. Uh,
mhm,
this system is based on lentivirus.
mhm,
So AV is also a type of virus, but it's much smaller, and
mhm,
they are DNA virus. So it's like a capsid, protein
mhm,
capsid, and then like DNA inside. But lentivirus is like a nanoparticle, like a lipid nanoparticle.
mhm,
And then like its viral genome is an RNA. So
mhm,
like the it's bigger. So for example, AAV is 20 to 30 nanometer in diameter,
mhm,
and lentivirus are like 100 nanometers. So
mhm,
the lentivirus can deliver something longer because the AV's like package capacity is only 4.7 kilobases. So for example, like if we want to deliver factor VIII, the DNA sequence is, even if we make the B domain deficient, it's still like almost full. But like with lentivirus, we can have something longer, so it's available for factor VIII. So uh, uh, adeno-associated virus is also like virus, but it's not mature virus because all the virus tool we use in the lab, they are already after bioengineered. So they can only deliver stuff to your cells, but cannot do any like self replicates. So won't be like influenza or something. It's just like
of course.
a vector for delivering stuff.
Mhm.
Yeah.
Um, so what is the biggest technical challenges with the AIDV?
Mm, there are many actually. So at the beginning, it doesn't work at all. So I did a lot of work to make it work finally. So I feel like, because for the AIDV, I said here, we need to pack three components in one particle. But
Mhm,
like the final product, I don't think they are gonna be 100% with all the three components. Maybe
mhm,
some of them only have the uh, template. Maybe some of them only have the protein. So, and also like they have like a computation because it's a nanoparticle. It's a nanoparticle anyway. So if you have too many, for example, Cas9 protein, then maybe it's hard to load enough DNA template donor for encoding the factor VIII sequence. So need to balance this amount to make sure both of the or all of the components are there. Uh,
mhm,
so this is like the first barrier, and the second one is like the effection or transduction, I would say, effectors ability for the nanoparticles, because after the modification, it cannot be as robust as, for example, COVID. Like if COVID, it can affect the cells very fast and very easily, but after bioengineer, maybe they cannot get into the cells so easy. So I need to do a lot of optimization for the outside. You can just imagine it's coronavirus, corona part. So if this part are not good, then they cannot go into the cells efficiently. So even if like my gene editing part for the three components are good enough, but if they cannot be successfully sent into the cells, like still it won't work. And also it's there are a lot of different types for the capsid or envelope protein. So
Mhm.
maybe some of the type are good for T cells, some of the type are good for neural cells, for example. So, but I think our system can work for hepatocytes, which are liver cells, because most of the nanoparticles prefer to go to the liver. So this is also why, like for the AIDV virus-like particle, liver is a very good target to do the gene therapy. So hemophilia. Mm,
Mhm.
yeah.
So you said that they prefer to go to the liver. Can they go to any other part of the body?
Yes.
Uh,
They can
I-
also,
mm.
mm, sorry.
Yeah, particularly I was thinking about the sex cells.
Oh, which cells? Sorry.
Sex cells, the reproduction.
Uh-huh.
It is a risk of
Yeah, yeah, yeah.
inheriting.
The, I think for reproduction cells, they are not that easy.
Mm-hmm.
'Cause like in our current lab, we did a lot of research about the biodistribution after injecting of this kind of virus-like particles or similar nanoparticles, lipid nanoparticles. Most of them just go to the livers, and then they also prefer to go to spleen. Um, but
Spleen?
spleen.
I don't understand.
Spleen is like the organ
Okay.
generate a lot of immune cells and also blood cells. It's close to stomach and the
Ja,
Mhm.
mjälten,
Ah, okay.
spinn, eller ja,
Okay.
förlåt, referens.
Nej,
Thank you, Therese.
det är ingen fara.
Hjälte. Mm.
Yeah.
Mm.
So
Mm.
like for other organs, they are actually very low. So we have data to support this. And also like in my gene editing systems, my factor 8 gene or other genes, there is no promoter. Promoter is like an element you can drive the gene to express. So if you don't have the promoter, the gene will not express. Even if you have the sequence there, it won't work. So for my case, there's no promoter for the factor 8 gene, and only after insert after the albumin, then like it can drive expressed by the albumin's promoter. So that's how it expressed. So just for example, if unfortunately this particle go to somewhere else, for example, like sex cells, and then because the sex cells they don't produce albumin,
Mm.
only the liver cells they can generate albumin. Like in other cell types, they won't express.
Mm.
They won't express. So even if there is a little bit leak, because there is no promoter of itself, there's no artificial promoter to drive this factor 8 expression. So only in liver knocking it can work. Other organs it won't work.
I
Most.
see.
Mm. So this is also why we want to do the gene insertion, like precisely insertion by CRISPR-Cas9 instead of the AAV thing I showed there with a promoter. Mm.
Mhm.
Yeah.
Um, so um, it's a more general question than about CRISPR,
Mm.
but just as anything that can be used so generally, um, are there any ethical concerns um, uh, that you have to deal with?
Uh, for example, CRISPR-Cas9, if we just do some texts in cell lines, like the cell lines we normally use in laboratories, or like use, some animal models like mice with some ethical permit, then
Mhm.
it's okay. Of course, we should not use it for gene editing to fix, for example, embryo, to change the baby's genomics. It's definitely not allowed, or it's not
Mhm.
legal. But mostly for therapeutic purpose, we just do for example, liver cells. It's not like you change the whole body's genome or just to improve your gene or whatever. It's
Mhm.
basically just for like save patients' life or improve their life qualities. So like under, because we need to do all the experiments under the, like after we got the ethical permit. So I think it's okay if we don't do something like embryo CRISPR editing, then that's insane. So for the therapeutic purpose, it's okay, I think.
Yes. So now we actually got a question from the audience. Um, can you predict when this therapy might be available?
That's a very good question, very critical to questions. Mm, honestly, I don't know, because I think for mice, I can do it very fast. But if you want to make it to just really inject to patients, then it's quite long way to go. Uh,
Mhm.
and honestly, I don't think it's like impossible or you can make the patients die immediately. But you always need to be very careful for patients. Actually, like during my PhD, that's like at the beginning of my PhD, it's like 7 years ago. So we did the hydrodynamic injection for the mice. At that time, there were patients in China. They were asking us, "Oh, can you use it for human being?" But at that moment, we don't have the AIDV particle. So, because you can inject the mice from the tail with very like high volume or whatever, but you cannot do that for human being. Uh, but like in Karolinska, we have the ATMP facility and GMP facility. These are for clinical translations. So I'm currently doing the optimization in the lab, and then hopefully we can, later, because my PI, he is also a clinical doctor, so maybe we can get some chance for some clinical trial, or maybe some collaboration with, for example, companies, so can, for example, purify our virus-like particles, so make it safer and more efficient, and then we can give it to patients. I wouldn't say how many years specifically, but I think in 5 years or,
Mhm.
but it's hard to say. But there are many other research groups doing similar stuff. Some of them use AAV vectors, but different system. And I think there are some, especially for hemophilia B, there are many clinical trials. Hemophilia A as well. Uh,
Mhm.
Yeah, so I think it's hopefully because there are many examples already. Now we change to a better, hopefully better system to deliver.
Mhm.
I hope that can answer.
Yes. Um, so, if we compare to a gene therapy that is already is already available, um, at least medically approved,
Hmm.
um, there is this concern that over a long time it tends to decline. Of course, we don't know how much it would decline because we will need
Yeah,
decades
yeah.
research to know that. In the best of worlds, gene therapy would, of course, be permanent. Um, uh, can you comment on if there is any difference with the AIDV compared to the existing gene therapy?
Uh, Yeah, it's a very nice question. So I think compare like our system, most of the current gene therapy, they only do, for example, um, gene knockout or gene knockdown, or I would say very simple change of the genome, because mostly we just use a Cas9 protein and I got guide RNA. So they can make some changes, but very small changes for the DNA. And then if we want to do something, but most of the genetic disorders, they are very complex. There are a lot of mutations. So like if we just change one or two DNA bases is not enough. So we want to do some I didn't talk about too much here. So our gene editing is actually homologous direct gene repair. So it's like a DNA repair pathway in all the cells, basically all the cells. But we need a donor plasmid, a donor DNA sequence for this. So uh, so this part is actually harder. So if I just want to deliver Cas9 and guide RNA, that's easier, which is also like most of the current, probably like very advanced or in clinic gene therapy. But if we really want to do something like gene insertion, then we need this DNA part as a template. So the advantage of our AIDV system is like we can pack all the necessary components in one or at least one formulation of the nanoparticles. So it's hard for other delivery system. For example, lipid nanoparticles, LNP, they can only pack RNA. I think DNA are still under researching, but hard so far. Extracellular vesicles, they can only pack some proteins and very little RNA DNA cannot have something so long like factor VIII, for example. AV, adeno-associated virus vector, as we mentioned, they can only pack DNA. So if we want to pack them like bigger things, like a proteins, then it's very hard. So there are many research, they try to encode the protein outside of the AV, but most of things are under like testing.
Mm.
So I think for our system is uh, mostly like all in one. So
Mm.
you can pack all the things in the same nanoparticles. And also this one is based on lentivirus. So actually it might be safer. For example, if compared with AV vectors, for this system, it should be less like immune response.
Mhm.
But for the gene editing part, because if you want to do gene editing, you need to break it first and then repair it. So for this part, it can get some toxicity, but we are also trying to do the modifications, focusing on this part. So there are many branches ongoing. Yeah. Um, I hope can
We
answer.
can follow this with interest.
Yeah. Many things to read as well.
Mhm.
Mm.
Yes. Um, so as a final question, you are originally from China.
Yeah.
How come that you decided to conduct research about this in Sweden? And um, what's your uh, what's your plans for your future career?
Yeah, for the future career, of course, I want to continue in Karolinska. It's an awesome platform. And also, thanks for the funding. So I can support my own salary and also like, because all the lab stuff cost a lot of money for sales, culture, whatever. So can use the money to support the research. So I'm really grateful. Actually, I came to Sweden just like I finished my PhD and was looking for jobs. And then I saw our lab and it's a very interesting project. So I just applied, got the offer, and then I came. So actually, the very good point of my career like line is like when I was doing my PhD, I just focused on the like upstream of the DNA like genome editing, but I focused on the the DNA thing. So I established some experiments or like knowledge for how to design, how to editing the DNA to make sure no mutation or make sure there's no error or make sure the gene can produce functionally. And then like my current lab, like we are actually an extracellular vesicle lab. So for this lab, we focus on delivery. So this is a very good combination if I can combine my experience for genome editing with delivery. Hello?
Uh,
Sorry, I feel
Sorry,
like it's a little
I
bit.
I think I lost connection for a moment. I didn't hear the last part. Say again.
Sorry. So I was just saying like during my PhD, I established some experiments for the genome part, but my like postdoc lab is like a delivery lab. So
Mm-hmm.
focus on the
I
virus-like
see.
particle. So if I can combine them, then I can deliver the gene editing tools like with our current labs, the technology for delivery. So hopefully we can achieve
Mm-hmm.
something real for patients like gene therapy.
Mm-hmm.
So this is like can be a mm, uh, something good for the patients.
Mm-hmm.
So so just came and after like when I just finished my PhD after this graduation, I was actually also very confused. But after uh, the life and the work in Sweden, now I'm enjoying my work and the life very well so far. So I hope I can continue my career. And of course, it's very meaningful, I think. And also like with this um, system, not only for hemophilia A, we can also use this system for other genetic disorders, hopefully. Yeah.
Mm-hmm.
Thank you.
Thank you. That was very interesting, and as I said before, we are very happy to hear
Thank you.
how it will continue. Thanks to you again uh, for conducting this very important research. Thank you for taking your time to present today. Um, and thank you to the audience for listening. And yes, with that, I close the session.
Yeah. Thank you. Bye.
Bye.
Good night.